Boa noite, bem-vindos, guten Abend. My name is Simone Lackner, and I'm doing a PhD in neuroscience here at the Champalimaud Foundation. And it's my pleasure tonight to guide you through this event where we will think about the gut. I just need to get the pointer. Well, now standing here on the stage, I must admit, looking into this very full auditorium, I feel a little bit butterflies in my belly. Maybe because I'm a tiny bit stressed. How many people of you can actually feel with me? Did you feel that before? Raise your hands. Yes, thank you very much. I'm not alone with that. And exactly because out of this reason, I was thinking, every time when I'm stressed, I feel nervous in my belly. So I wanted to learn more about the brain and the gut and how they are communicating with each other. So I was digging into the scientific journals and scientific literature to learn more about this interaction. And a completely new world was revealed to me. And together with my partner, Marta Camacho, she was my partner in crime, we decided to bring this world of the unknown tonight to you. So let me start with some basic ideas you maybe remember. Our human body is built out of many different cells. Many different cells who have many different properties. And one of these human cells, well, every human cell, has a full, complete human genome, which you get from your parents, half of your father and half from your mother. And this human genome has the complete instructions to tell the cell to what it should develop and which organ it should be part, for example, of the guest of honor tonight, the gut. But did you actually know that most of the cells in our gut are not even human? Well, for a fact, they are microbial. And not only in our gut, actually microbes are all over our body. And the sum of these microbial cells, scientists call the microbiota, the human microbiota. So every single cell of these microbes come with its own genome. So we are actually outnumbered by all these genomes from these microbes, which the scientists call the human microbiome, which is an analogy to the human genome. As all these cells come with its own genome, it also has different functions, most likely different functions to our own human cells. So they enhance us kind of in our function. But what does this mean for us as an individual and human in this ecosystem we all live together with microbes, with animals, with plants, well, everything basically what lives. The borders become a bit blurry. So Joanna Rico, our invited bioartist, who has also an exhibition outside, made this really nice piece of art. And when you look at it, it might strike you that our human body, together with human cells and microbial cells, is an ecosystem by itself. So let's look again at the microbes in our stomach. They are really nice helpers. So they help us, for example, to digest these little bits of food, like fiber. Fiber is actually very important for us, and most of the things we eat contain fiber. And it's very difficult to digest. Without those microbes who actually can digest this fiber and give us all the nutrition we cannot extract from it, we actually have an enhanced function. But even more, these microbes help us also to metabolize drugs and shape our immune system, because our immune system has to learn which microbes are good and which are bad. Recent studies actually suggest even that these microbes are involved in changing our behavior. And our guests today, José Oliveira and John Cryan, will tell us more about that. So one question I had was, where do these tiny microbes come from? Well, we initially start with the most sterile gut, which then accumulates microbes during and after birth. And this initial composition of microbes depends a lot on how we were actually born, which can be vaginally or by C-section. But then further on, these microbes develop, and that depends a lot on how we get fed, which can be either breastfed or bottle-fed. But only after introduction of solid food, we will start emerging or accumulating more of those microbes, which we also find in adult guts. So when we look now at the microbial composition of this infant, we can see that the diversity and the stability of the microbial composition is still very low. And it can change from day to day as the baby gets exposed to all these family members, to different food and different environments. That changed way more than after a year or two, when the kid already eats more solid food, plays with different children, and its microbial composition becomes already more stable and diverse than in the infant. Then when we get into this age of young adults, we party, we run around, we have our experience, we travel, and do maybe the one or other diet experiment. And that's the time where actually our microbial composition becomes the most diverse and stable. The peak, though, is then when we become more settled, like we become the adult. We stabilize our life, we maybe build a family, we buy a house, we don't run around anymore that much. And that's the time where our microbial composition becomes the most complex. And most likely it will stay like that for the rest of the life. Although some research has shown that once we get into this really old age, where we have aches in the leg, in the arm, and the head, and we have to take a lot of different medication, also our diet change, and we maybe don't leave anymore our house or travel around, that is where our microbial composition loses on stability. And that can happen over day from day and week to week. So when we're looking now at this plot as a summary, I think we can see quite well how age has an impact on our microbial composition. But so does diet. So scientists were looking at the different dietary habits of people and their microbial composition. And what they saw is that people who eat mainly animal protein and saturated fats had more of these bacteria. But people who tended to eat more carbohydrates and simple sugars had more of these prevotella bacteria. And this pattern was also true when they looked at people across nations and culture. So this is a very abstract representation of all these individuals which were participating in this study. And scientists can then look selectively to this prevotella and bacteroids bacteria. And what they saw is that people like Africans from Malawi who eat more corn and Amerindians from Venezuela who eat more mandioca fall more in the category of the prevotella bacteria, while Americans, so also Europeans, who eat more processed food and meat fall in the category of the bacteria. So in summary, we can see that our choice of dietary habits also influence the microbial composition which we have in our gut. So I think you hopefully got now a little view into our inner ecosystem and how these microbes are having are influenced by our diet and by our age. But why I'm here today, and hopefully you also, is I wanted to understand how these microbes are actually changing the behavior in our brain. Well, one thing is for sure that the nervous system connects our brain to all the organs and everything what happens in our body. But when things are going really wrong, then there's a very, and the gut has to communicate very fast to the brain. There's one nerve which is called the vagus nerve, which provides the most rapid and fastest connection. When things can happen on a more lower time scale, like slower time scale, like via chemicals in the blood, we have the hormone system. And last but not least, as we talk here about microbes all over our body, of course, the immune system has also a very important role. So to understand now all this interaction, the brain and the gut and the microbes, we invited the experts of the field tonight for you. And we will start with John Cryan, who will talk about the interaction of the brain and the gut and the microbes, and how impacts like stress and neurological disorders can change that. And then second, we have José Oliveira, who will introduce us to the field of immunopsychiatry and immunogenetics, and he will tell us how microbes can influence our mental health. So with that, I want to thank you for showing up, and I wish you all a very educational evening. And yeah, that's for me it, and I would like to have Marta Camacho now to come on stage to introduce our first speaker. Thank you very much.
All for coming. I am a grateful representative of an amazing and big team of scientists who are from the Champalimaud Neuroscience program, but also from the ITKB, who spent hours preparing this event. I hope you learn and enjoy throughout this evening. All of this was done in scarce few time. No science was hurt or procrastinated because the big boss is here, so it's a good thing to say. It's been a pleasure to organize, and I would like to invite our first speaker, Professor John Cryan. He's professor and chair of the Department of Anatomy and Neuroscience in the University College of Cork, Ireland. He is a PI at the APC Microbiome Institute, and he's a prolific neuroscientist with over 250 scientific papers published so far. I think one day he'll have more scientific papers that we have microbes in our guts. I want to thank his presence, and we've been learning with him and other distinguished scientists in this advanced course that we have had in the last couple of days here at the Champalimaud Foundation. And he has often stressed the importance of contextualizing these new results in an evolutionary approach. And he keeps telling us how important it is to notice that the microbes were there first. Well, I think in a very short future generation of scientists, whatever we study in the microbiome, John Cryan was there first. John, please welcome.
Thank you very much, Marta, for the very kind words and for the invitation to be here. Thank you so much to Simone for doing such an amazing job in introducing a field that you don't work on. So I think we need to give Simone another round of applause for really, it's testimony to your institute here and your foundation that you have graduate students that are so easily able to jump into fields outside their own comfort zone, and for Marta as well to really bring this evening together. So I hope it's worth it. It's a great crowd, and over the next 20 minutes or so, I'm going to share with you the journey that we've been on in Cork in the south of Ireland, in trying to really tease apart how you really have a biology to your gut feelings. That when you have these gut instincts and feeling a bit gutsy about being gutted, that there is actually a relevance to it. And so it may not just be the butterflies in your tummy, but that we all feel, but it may be something more physiological. And it really has been a journey that we've been on. And if someone told me 10 years ago, I would like to think that I would be here at a wonderful neuroscience institute, but talking about microbes would have been seen as being out of this world. And because in neuroscience, we tend to focus on everything above the neck. And so I'm taking you on that journey, on the whole body idea of what we're doing, and hopefully I can convince you of the relevance. So my story begins because I have young kids, and my 5-year-old's favorite bedtime story is The Adventures of Pinocchio. And you all know this story. You have a puppet that is sculpted by the puppeteer Geppetto. And in doing so, Geppetto is essential for Pinocchio in his early life. But as Pinocchio goes off and has some mad adventures, Geppetto is always there, kind of keeping an eye on him. And over time, over the last number of years, in a way, that's kind of what I've begun to see our microbiome. The bacteria within our gut is helping our brain keep out of trouble and have many adventures. And so hopefully I can convince you that in the next while. And so where does our story begin? Our story begins in, I like to begin it in the 1820s in rural Michigan in the US. And there, at that time, we knew very little about how the digestive system worked. So you have to think about that. And there you had a US Army surgeon named William Beaumont. And Beaumont was a very curious clinician scientist, very typical of the clinician scientist. And he had a patient called Alexis St. Martin. Now, St. Martin was a Canadian fur trader, and unfortunately for him, he had a gunshot wound to his abdomen. So guns were a problem in the US even back then. And Beaumont was able to keep him alive, and he survived this, but a hole or a fistula remained. And so Beaumont said, well, now I have a real window, literally, into the digestive system, and maybe I could perform some experiments. And this was before ethical committees really got into their major role. And so he kept St. Martin as a human guinea pig for many years. And basically started doing experiments on food through the mouth and put food into the actual wound and looked at the gastric juices and basically wrote about how we digest food. And wrote about it in this very seminal book. And this is the foundation of modern gastroenterology. And really important findings from this. Now, why am I telling you this? Well, one of the things that he wrote about was that when St. Martin became angry or irritable, and it's perhaps not so surprising if someone is shoving a lump of corned beef regularly into your abdomen that you might be somewhat irritable or frustrated, and that the rate of digestion changed, that your emotions changed your gut function, that we have this brain-gut axis. And that was very important for our understanding of where this begins. With the advent of brain imaging in the 1980s and subsequently, we began to really realize how this is a bidirectional communication. That if you stimulate the gut, you can see in the brain that it lights up in key emotional areas. And we have this brain-gut axis, and there are many routes of communication, and it is vital for what we call homeostasis, maintaining normal physiological states. Of these routes, we have the enteric nervous system. I just want to mention that because this is the nervous system within the gut. There are more neurons in your gut than there are in your spinal cord. It's often very much neglected in neuroscience, and it's also called the second brain. And it doesn't just play a role in motility, but plays fundamental roles in maintaining homeostasis. We've all heard from Simone about the vagus nerve. That's a key player, and I'll come back to that a little later. So how did I get interested in the gut? Well, my lab is interested in stress. And we're interested in how stress, traumatic stress, or chronic stress affects the brain and affects the entire body. And it's worth pointing out that we are living in a very stressful world. I was in a taxi in Lisbon today, and it wasn't unlike that. My desk doesn't unlike that at times. And, you know, all of these, we've come through a financial crisis in Ireland. We are part of the pigs as well. And so we all know about it. And so there are lots of stressors on our daily lives. All we have to do is just turn on the television, and we even get more stressed about what's going on in the world around us and how this can have a major impact on where we're going. And Hans Selye is the father of stress research. And he has a famous saying about that, it's not stress that kills us, but it's our reaction to it. And that's really what informs my research, is why on this roller coaster of life, if two people are subjected to the same stressors, that some will be susceptible and others will be resilient. And a lot of focus over the years in stress research has been on the negative aspects of stress. What are the factors that make some people more susceptible? But over the last maybe 5 years or so, there's a growing appreciation trying to look at resilience. And what are the factors that make some people more resilient than others? We know that genetics plays a role. We know that the growing field of epigenetics plays a role. But what if it's got to do with something in your gut? What if it's got to do with something in the microbes of your gut? And that's kind of where we've been over the last number of years. It's also worth pointing out that stress is a whole body syndrome. It doesn't just affect a few neurons in the hippocampus, and we love these neurons and we study them heavily, but it does affect the entire body, affects the endocrine system, the hormonal system, the immune system very much, and locally in the gut. In terms of what's going on in the gut, one of the key things that stress does, it makes gut barrier function more leaky. And we all hear a lot more nowadays about these leaky guts, which allows for the immune system to be activated, and you have feedback to the brain that way. And as a basic scientist, we require animal models to try and study these syndromes. And the problem in stress-related disorders, and it's worth highlighting that, is that there are difficulties in trying to model complex human psychopathology in animals. And I like this Gary Larson cartoon, not just because it is an Irish setter, but because it really exemplifies the problems that we can't just put our rat and our mouse on a couch and ask it about how its parents treated it during its childhood, or whether it has constant thoughts of death or suicide. But what we do know is that the key circuits in the brain that underlie many of these stress-related psychiatric disorders have been conserved. Through the amygdala fear circuits, through the striatal reward circuits, through the hippocampus, some of the cognitive circuits, and so that we can use rats and mice with some level of confidence to tease apart mechanisms underlying how stress affects the brain and how stress affects the body. And a number of years ago, Siobhan O'Mahony and Ted Dinan and myself showed in a model of early life stress where rat pups were separated from their moms from postnatal day 2 to 12, that when they grow up, that they had a wide-scale change in physiology. They had changes in their immune system, changes in their endocrine system. They had increased visceral pain. They were changes in their gut barrier function. But one of the things that Siobhan showed in that paper was that they had differences in the microbes in their gut, in that there was a narrowing of diversity in these animals in adulthood, that there was a signature in their gut of stress that happened a long time earlier. And that started us thinking about, well, what could this mean? We've recently replicated this in a model of prenatal stress. And human studies are emerging in moms that have high stress during pregnancy, that the microbiome of the infants at 3 months is different. And so a lot of this is actually becoming translational. And so that got us thinking, what is this microbiome? What can it do? And can we harness it to try and understand stress? And we've had a lovely introduction already, so I won't dwell on this too much, except to say that it is illegal to give a talk without telling you that we are 99% microbial in terms of genes. So all of the money that has been spent on the Human Genome Project has been really for just 1% of our genes. And so that's humbling. The other humbling part to remember is, as a neuroscientist, is that the weight of your microbes is about that of your brain. It's about 3 pounds. So again, humbling. If I'd given this talk a few months ago, I would have said that we are about 10 times more microbial cells than human cells. But this is a field that is changing daily. There are papers coming out in high-profile journals all the time. And so we have to, keep up with it and keep up with technology. So a paper came out in Cell in January, which basically questioned that very heavily using neurotechnology and other mathematical modeling. And basically now we can say that we're about 1.3 to 1 more microbial. So we're still more microbial, but just about in terms of cells. And it means that every day, when you go to the bathroom and you get rid of some of your microbes, you are slowly becoming more human. So we've already heard about where do we get our microbes from. We're largely thought to be sterile in utero, although the placenta has been shown to have a microbiome, and this has been challenged more and more. But we do get our microbes as we emerge through the birth canal from our moms in early life. So yes, it's another thing in psychiatry we can blame our poor mothers for. And there are many, Simone has handled most of these in different ways. Mode of nutritional provision is very important. Gestational age, preterms have a different microbiome. Genetics plays a role, even though I did poo-poo the human genome, it does play a key role with about over 40% of our microbiome being heritable. Hospitalization, antibiotics, the environment. You know, in this amazing building here, the architecture is really integrated into the workplace. And so the built environment is very much alive in this foundation. But there's a whole new science emerging in the built environment, which is how does the microbiome interface with the built environment? And a lot of exciting work emerging there, interfacing architecture, engineering, and microbiome work. And even bringing that one step forward, people are beginning to look at this in the context of mental health. And can we design better buildings for our microbes to influence our mental health in a better way? And this is just starting. But also in terms of the environment, living in a green environment versus living in a city will change your microbiome. I've already talked about how maternal stress, but also maternal infection will play a role. And we do know that antibiotics, especially in early life, will have long-lasting, enduring effects, at least in some aspects of metabolic function, and that's becoming more and more clear as we get more and more evidence. So we're quite interested in aging, and the whole idea of microbiome and aging goes back to this guy, Eli Metchnikoff. Now Metchnikoff won the Nobel Prize in 1908 for his classical work in immunology in discovering macrophages and phagocytosis. And what happened was that later in his career, as does happen, some scientists, they come up with some crazy ideas later in their career and they're mostly ignored. And people say, weren't they really good earlier on in their career and made all these wonderful findings? But what Metchnikoff was interested in was why some people age better and live longer. And he focused on people living in Eastern Europe, in Bulgaria. And he realized that they ate a lot of fermented food containing lactic acid bacteria. And he's written about it elegantly in these studies and basically put forward the idea that the microbiome is central for our longevity. And we're interested in the microbiome in aging, in the perinatal period, as I showed, and also in adolescence. And what we want to do, really, is try and place the gut-brain axis at the center of what is going on in the aging process. Now, Metchnikoff was ignored for 80 years or so because people didn't think there was any basis to what he was looking at. But now, with microbiome technology advancing, we're really beginning to see that the microbiome is playing a critical role in how we age healthily. Studies from my colleagues in Cork, published in Nature a couple of years ago, showed that taking 180 aged people and looking at their microbiome and looked at their health outcome, they showed a clear link between the microbiome and health. The more diverse the microbiome, the better the health outcome. Moreover, what they showed was what was driving the diversity of the microbiome was the diversity in their diet. And so we have a diet, microbiome, health outcome. Now that was in aged individuals, but that also is important for all of us in this audience. And if one thing that I can tell you today is that while you can't change your genome, you can try and change your microbiome. And that's one of the key things that our diet is able to do. And so we're very interested in understanding this microbiome, gut-brain axis in aging, and understanding how stress has direct effects on the aging brain and how this may lead to some of the behavioral negative outcomes that we see in the aging process. And we're slowly beginning to understand that diet and nutrition is a great way to intervene, as are certain what we call probiotics, and that these can obviate maybe some of the effects of stress and be able to influence both our metabolic health as well as our mental health and our aging over our lifespan. So how do we study the microbiome in the lab? What are the types of experiments we do in a basic science way? Well, one of the easiest ways in biology to study something, and also in all science, is to get rid of it and see how the system operates. And so we have, and we're lucky to have one in Cork, we have a system called germ-free animals. These are animals that grow up without any microbes. Now, the concept of being germ-free has, people have been amazed by for a long time. Here's a sci-fi magazine from 1927 about a germ-free man. So, but we can have germ-free mice, and in the 1950s and 60s, these emerged. And we've been profiling the behavior, and we've shown that there are clear neurodevelopmental changes in these mice that tell us that we need bacteria in our gut for normal brain development. I'll share you with a study that we just published last week, which showed that when we looked at genes that were changed in germ-free versus control animals in the prefrontal cortex. Now, this is an area of the brain involved in decision-making and executive function and planning, and also in relation to regulating the HPA axis, the stress response. And we found a large-scale number of genes that were upregulated. But intriguingly, a lot of them, 14% or so, were involved in the process of making myelin. Now, myelin is like the insulation on nerve cells. It keeps nerve cells communicating appropriately. And you may know of it in the context of multiple sclerosis and other demyelinating disorders where there's loss of myelin. And here we've shown for the first time that how myelin is regulated in the brain is very much driven by having microbes in your gut. When we looked in the microscope, we found that there was an increased myelination in the prefrontal cortex of these animals. So again, telling us that fundamental brain processes are regulated by the microbiome. Also, we found that these changes tended to be much more common in males than females. And we saw that in other parameters that we looked at. Now, I'm a stress researcher. Unfortunately, stress-related disorders occur more in females than males. So it was a slight conundrum, but there are brain disorders that are more male-specific. They include neurodevelopmental disorders like schizophrenia and autism spectrum disorders. So we were intrigued to see, well, how did these mice behave in behaviors relevant to autism? And we looked at sociability in these mice. Now, if you put a mouse in a situation like this, where it has the opportunity to spend time with another mouse or an empty chamber, mice, like humans, are quite social. So they'll prefer to spend time with another mouse, but not if they don't have microbes in their gut. The same, mice, like some humans, are a little bit fickle. So if you give them the opportunity to spend time with mice that they already know or a new playmate, they'll gravitate towards a new playmate. But again, not if they're a germ-free mice. And so that allows us to start thinking that some of the core symptoms of autism are very much dependent on some of the behavior, on them having appropriate microbes in the gut. There are other models of autism that have shown changes in the microbiome, and it's a very emerging field, although a lot of very controversial field overall, and a long way to go to try and dissect out the mechanisms. It allowed us to start thinking about it, well, why would we need microbes in our gut for normal social behavior? And this is what Marta referred to. We started thinking up a kind of constructs. Well, what's in it for the microbes? You know, if we are in a social environment, you can start thinking that microbes could actually, pass on from one to the other in a much easier way, and it would increase their fitness. But I collaborated with Seth Bordenstein in Vanderbilt, an evolutionary microbiologist, and Seth basically told me, John, no, that's the wrong question, because you have to remember the microbes were there first, and that we have evolved, and our brains have evolved always with microbes there. And so it's a coevolution, it's a hologenome effect, and so we can't neglect things. So that's the other take-home message I give you. Always remember, microbes were there first. So how do microbes signal to the brain? Well, Simone mentioned some of the mechanisms, and we're just slowly working them out, and it's going to inform my lab, and we have a lot of people working on this right now, and it's a very exciting area, a long way to go to really tease it apart. But what we do know is that we're beginning to understand in neuroscience, especially the neuroscience of psychiatric illness, we're beginning to really understand what's going on in the brain and how this feeds into predisposing to certain mental disorders. But now we have to juxtapose what's going on in the microbiome at the same time, or even way earlier, to try and integrate this into a clear story. So I told you that stress, especially in early life, changes the microbiome. I also told you that mice lacking the microbiome have an exaggerated stress response and altered neurodevelopment, changes in anxiety, cognition, and social behavior. I didn't go into detail on this, but just to reinforce, these are the key findings we have in our germ-free animals. And that led to the question, could modulation of microbiota have beneficial effect on brain health? And so we fed mice with lactobacillus for a number of weeks and then tested them on well-known behavioral assays. This assay here is called the elevated plus maze. If you put a mouse in this assay, it'll choose between an open and a closed arm. And if you give the mice like the closed, secure areas, and they'll stay in there. If you give that mouse Valium, it'll come out here. What happened was when we gave the mouse the lactobacillus, it behaved as if it was already on Valium. Really striking findings. This is a forced swim test. Mice are good swimmers, but they don't like to swim, they'll give up. But if you give them antidepressant drugs like Prozac, they'll keep swimming. And our animals behaved as if they were on Prozac, those that were given the lactobacillus. When we stress them, you have a normal stress response here in terms of hormone output, but when they were fed the lactobacillus, they are much more chilled out and relaxed. And so we have a way of changing behavior, physiology, and when we looked in the brains, we looked for GABA, which is a neurotransmitter and what Valium works, and we saw wide-scale changes in GABA neurotransmission. So this was really, really cool and really exciting data because these were normal, healthy animals. And the question is how? How can microbes in the gut signal to the brain and have such widespread changes? We've mentioned the vagus nerve, and so that's giving it away. So together with John Bienenstock's group in Canada, we performed vagotomy and cut this communication, this neural communication between the periphery and the brain. And to make a long story short, all of the effects that we had seen were gone. So this tells us that what happens in vagus will really affect your emotions. And that this is one direct way of communication between the gut and the brain. It's not the only one, but it is one that is important. And this has allowed, this was a cartoon that the dreaded Daily Mail published in the UK when we published our paper, but it's also important to, as we move forward with this, to understand that not all influences of microbes are going to be positive. There's going to be some negative effects as well. No microbiome talk is complete without talking about fecal microbiota transplantation. Taking someone else's poo. Would you do it? Would anyone do it? One? A few brave people. Well, I can tell you, you would do it if you had Clostridium difficile infection. C. diff is a killer. It's a hospital-borne infection that people who are retractable to treatment die. And so in a study a few years ago from Holland, it showed that if they took a fecal transplant, that there was a 90% success rate. Now in medicine, there is no such thing as 90% success rates in other fields. So it has changed our whole perspective of what a medicine is. There are newer, more aesthetically pleasing ways of delivering it in tablet form. These are often called crapsules. There's a project called Repopulate. This is changing our whole concept of modern medicine. But what about in psychiatry and behavior? Well, to my knowledge, but I'm afraid to look on YouTube too much, but there isn't any human studies as yet. But there is this intriguing study in animals from Steve Collins' group, where they looked at 2 mouse strains that had different microbiome and also had different anxiety behavior. And they did a fecal transplant. And what they found was that they were basically able to change this resilient mouse into a normal, into a stress-susceptible mouse, and vice versa. So if this could be recapitulated in humans, it has huge implications. Not least of all, if you ever did have to get a fecal transplant for C. diff, you really should get a good psychological profile of your donor, just in case. So a lot of what I've told you tonight in this talk has been about our basic science work, our basic neuroscience work, and our basic work in animals. But the field is moving now more to human translation. And together with my close colleagues, Ted Dinan and Catherine Stanton, we've coined this term psychobiotics. These are bacteria which, if taken in adequate amounts, will confer a mental health benefit. And we're beginning now to do human studies. This is one of our researchers, Andrew Allen, doing EEG on a television reporter in Ireland. And this is really changing how we're going. We're able to start to translate some of our animal studies all the way into humans and healthy volunteers, and hopefully, eventually, into patients. So I'll back to Geppetto and Pinocchio. And, you know, when I think about this, the question that always comes up is, who's really in control? And as I know also in my personal life, what I've come to realize is that the person really in control is often the wife or partner of the person who thinks they're in control. And so perhaps as the brain thinks it's in control, but that it might be the actual microbiome that is actually pulling the strings, and that is very important for how the brain is mediating its effects. In the 20th century, we focused all our efforts in microbial medicine and killing bugs, and that saved millions and millions of lives. But now we realize how important our microbiome is for maintaining health, and especially for maintaining brain health. And so into the future, this is going to be a crucial area, and that our state of gut will markedly affect our state of mind. Thank you very much. I want to just acknowledge my team in Cork, and Ted Dinan in particular, who I run the lab with. He's the chief of psychiatry in Cork, our industry collaborators, our funding agencies, and I leave you with this thank you from the microbiome. Thank you very much. And I will take questions.
So thank you, John, for the wonderful talk today. The audience, please feel free. You can talk in Portuguese and I'll try to translate to the best of my knowledge. You can also leave your talk till after.
So what would you say would be the ideal diet for someone that wants to have a diverse genome?
Yeah, so the studies to date would say that a diet that has a lot of green and a lot of fiber. Fiber seems to be quite crucial. And the studies emerging showing that we have, as a society, lost the ability to even digest some crucial fibers. That if you look at, as Simone said, in other populations, the Amerindians or in Malawi, they retain these bacteria that can digest fibers. So unless we want to stop that from occurring, we need to maintain fibers. There are certain things we should avoid. We should avoid processed food as much as possible. We should avoid emulsifiers, sweeteners. These have all been shown to have negative effects on our diet. And so, you know, diversity is the key in terms of what we're doing, and lack of processed food. That's as much as we really know right now.
Hello. Yeah.
Yeah.
You mentioned something about fermented foods.
Yeah.
I work with wine, and I would like to know if there's any relation to what you've been speaking about.
Okay, so I'm Irish. We are quite aware of the physiological effects of alcohol. The direct answer, if you work with red wine, then the red wine contains polyphenols. And there's a lot of work going into polyphenols, like resveratrol and others that are shown to modify the microbiome in somewhat of a good way. Alcohol, and there isn't an awful lot of data yet. We're doing some studies on the impact of alcohol on the microbiome right now, and are able to show some negative effects of alcohol through vapor in animal models. So it's kind of, you know, a push-shove. I don't think if I was recommending ways to get fermented foods and probiotics, I don't think I would be recommending wine as my first choice food, unfortunately.
Thank you.
I'm waiting for the Guinness one to emerge, but yeah.
So there's a lot of controversy about the stability of your microbiome in relation to diet. For example, like some studies say if you change your diet for 2 weeks, the microbiome changes. But this would, if it's so important for our health, it would be really surprising that these changes that the microbiota is so unstable, right? So what's your thoughts on how stable it is and how it's going to keep throughout life?
Sure. No, I think it's a great question. I think for the most part, the microbiome is relatively stable. There's a study. It's one from Eric Alm in MIT that I like to share with my students and postdocs, because it's an N of 2. It's him and his postdoc, and they basically looked at their microbiome every day for a year. Now that's a lot of a commitment because you get your microbiome from your stool, so you have to, and so they looked at stability over that year, and with the exception of when the postdoc went to Bangkok and got infection and was FedExing back and when Eric himself had to go on antibiotics, it was relatively stable overall, and there are lots of studies like that. But rapid changes in diets, like moving to a very high protein diet, has been shown in 24 hours to change the microbiome. What we don't know is, no one's looking at, does that modify mood or behavior? You know, people who go on these paleo diets will refer to having some changes in their cognitive performance and being a bit more grumpy and a bit more whatever. But you're also removing alcohol from the diet, so it's also maybe due to some of the effects, at least in Ireland. But you know, so I think we have to understand that. The study from Cork in the elderly was a 2-year prospective study looking at changes over time, and they also found that people's diet changed and their microbiome changed as they moved from living in the community to living in a nursing home, and this had negative effects on their health. So you know, we're beginning to interface that quite well, and I think generally in adulthood we see relative stability in our diets. But there's going to be a big inter-individual differences. That your diet will be different to the person beside you, but more interestingly, your microbiome is going to be quite different as well. Yeah.
Hello. Thank you very much for the talk and for the humor. Really nice talk. So you showed us that if you give a mouse probiotics, then he will show some behaviors that are considered good behaviors,
yep.
Right? And you also showed us that vagus nerve amputation leads to a decrease in that effect, or you get rid of it. Can the negative effects of having a bad microbiome also be ameliorated through either vagus nerve stimulation or cutting it off?
No, that's a great question, and we don't know. One of the areas that we've worked on this is looking at how certain side effects of drugs changes the microbiome. So antipsychotics are taken by 1% of the population, and they cause huge weight gain. And that weight gain, we've shown, it changes the microbiome. And we could reverse that by manipulating the microbiome. In this case, we used antibiotics to manipulate it, but we would hope in the future to use probiotics to do that, so we can get rid of some of the negative effects of an altered microbiome in that way. The stress effects on the microbiome, we've been able to show that area life stress changes the microbiome, and we can reverse that by supplementing with polyunsaturated fatty acids. So using dietary means to try and reverse some of these changes. So yeah, I think that this is an important area. And that what's going to happen in the future in medicine is people will have a diagnostic snapshot of their microbiome in health and follow it up over disease and be able to show what are the factors that they could use to modify it and bring it back to what it was normally. And that sounds a bit like science fiction right now, but it is going to become part of the diagnostic repertoire in the future.
Okay, and regarding the specific vagus nerve communication,
yeah,
there, like stimulation doesn't lead to improvement of.
We haven't done anything there yet, but there's no data yet. But I would imagine it probably should. Yeah.
Yes, thank you for your very stimulating talk, and. To increase a little bit the anxiety for knowing these problems. And what I wanted to is a very brief comment on the pathogenetic mechanisms of some disease that are thinking that are connected, like irritable bowel syndrome, the fibromyalgia, and all arthritis, this sort of arthritis that are very crumbling, and the microbiota. And also for some very important problem in our daily medicine, that is the use of the knowing how is the thrombotic state, because apparently the gut microbiota influence the activity of the platelets.
Yeah. So that's a huge question. So quickly, on thrombosis and arteriosclerosis, microbes make metabolites that are negative TMAO from meat, and so the diet microbial interaction that we now know predisposes to arteriosclerosis. So a lot of work going on in cardiovascular like that. And same in rheumatoid and inflammatory bowel disease, there is a large literature now implicating the microbiome in driving not just only disease susceptibility, but also flare-up in these diseases. With irritable bowel syndrome, we're doing a lot of work ourselves because it's a brain-gut disorder, the most common gastrointestinal disorder, and there we can actually treat some of the symptoms by targeting with certain probiotics, and there is a different microbiome as well. So across multiple areas of medicine now, we're beginning to see that we may be able to not only just use it as a diagnostic, but also as a treatment intervention, but we just need to find out much more about it. Yeah.
I have two questions. One is, I've read a lot about the ketogenic diet and the mood stabilization, that people, they don't have peaks. The effects on eating much more fats in the diet and in the brain. And the second question is, we're talking about food, but we contact with a lot of bacteria by touching a pet, eating dirt, like the little kids. That sterile environment doesn't put us in contact with those things. It's the combination. I would like to know the effect of that.
Sure. So the first question, a ketogenic diet, there is work going on. Zoltan Sarnay in Australia is working on this in the context of microbiome in relation to maybe having an impact on schizophrenia. There are some people beginning to work on it in the context of epilepsy. I've not seen much in the context of mood yet, but it definitely, anything diet will impact the microbiome in some way. So it definitely needs to be studied, but we need to move from correlation to causality as well. Regarding pets, there's some exciting work from Jack Gilbert and others where they've looked at it when people move house. What are the factors that are changing your microbiome and bring your new house to your old microbiome? And having a pet was one of them that really reinforced this much quicker. And they've done a lot of work about having dogs and the influence of that. And this definitely does agree with the old friends hypothesis of Graham Rook and Chris Lowry and the old hygiene hypothesis idea that we are living in a hyper clean environment, and that does narrow the bacterial diversity that we get exposed to in our environment, and that perhaps isn't a good thing overall, and that may also give rise to some of the reasons why we're having such autoimmune diseases.
Last question.
Okay. Hi. I have a question, which is this. Could these effects be mediated not by the microbes themselves, but by inflammation, so that it would change an inflammatory reaction? Because we know inflammation is implicated in so many different things, you know.
Sure, and we have one of the world's experts on inflammation, Keith Kelly, here in the audience, inflammation in the brain, and that's a really exciting area of psychoneuroimmunology over the last 30 or 40 years, and we know the immune system will play a key role of regulating the brain in general. But what I'm beginning to see now is that maybe it's the microbes that are regulating the inflammatory effect that is regulating the brain. Keith might not agree with me, but we're beginning to really tease that apart overall, and there's definitely a very close relationship there. But the question is, where does the inflammation start? I can tell you where the microbes start, but so it's getting at that point. And most of our inflammatory models are based on a microbial infection, so it's all linked.
Thank you for your questions. Please keep in mind that you can ask further questions on outside on the activities to both speakers. Let's please thank again Professor John Cryan. Okay, so now that we're all got brain access specialists, we're going now to complex a bit more. Ooh, I don't think that's correct in English. To add another layer of complexity to this topic and talk about mental health in human studies. So we've invited a C-section baby, José Oliveira, who was who was able to surmount that small microbiota step back early in life and have a very promising career. He studied in Coimbra to do his medical training. He then did his PhD in immunopsychiatry in Université de Paris-Est, and then he came back to Portugal to finish his residency in psychiatry, and is now collaborating with us at the neuropsychiatry unit here at the Champalimaud Foundation. So once I got to know him about 2 months ago, we came to the conclusion that we had been in Coimbra at the same time, shared friends, and even went to the same weddings and never met or talked. Now I'm lucky enough to call him a friend. Please welcome José Oliveira.
Thank you very much for this very warm welcome, and I'm very happy to see that this event is sold out. It is for me a pleasure to see that there's such a strong interest in this relationship between microbes and mental health. It is an honor for me to participate in this discussion, and my contribution will be on the interaction between the microbes and their host, in this case the human beings, how the immune system is implicated in this interaction and its associations with mental health, and specifically with some psychiatric disorders. I would like to start by stressing this idea that through evolution, microbes had an incredible capacity of adapting to virtually any single environment you can imagine on Earth, since the very hot spring waters in the Yellowstone Park in the United States to the freezing waters of some lakes like the Baltic Lake, or the deep oceans where they're submitted to very high pressures, it's unliving conditions, but they also colonize other organisms like us, and this starts since very early in life, which is a key period, and this relationship depends on many, many factors. Some of them were already referred. It depends on genetic factors, on maternal health, on the method of delivery. It might have affected me because I'm a C-section baby, subsequent nutrition, maternal and postnatal exposure to antibiotics, and the immune system. So we can think of ourselves as a super complex ecosystem, and this is an ecosystem also because we live in symbiosis with these microorganisms. And symbiosis means that we both profit from this relationship, and evolution favored the symbiosis instead of a simple parasitism. So I want to add to the previous talks, which were excellent, but we profit in terms of digestion and in terms of self-defense against infections because they help better shape our immune system. I will give you an example of bacteria living in our system because this symbiosis is in fact a very complex diplomacy, and many rules have to be more or less respected. This, I don't know if you experimented the activities, the staining activities upstairs. If you didn't, you can try after the event. If you stain this bacteria using the gram staining technique and observe it under the microscope, it won't stain, so it's a gram-negative bacteria. And this kind of bacteria, they have an essential component on their bacterial cell walls, which is called lipopolysaccharide. Most of you probably heard about it. And in the environment where these bacteria are present, lipopolysaccharide is a common contaminator, we may say. This environment can be the gut. And the intestinal cell wall is a barrier that keeps the diplomacy and these rules are respected. But in some conditions, like psychosocial stress exposure, namely childhood trauma, for example, or life adversities or a stressful lifestyle, but also intestinal inflammation in the case of some chronic inflammatory conditions like Crohn's disease, or even dysbiosis, which is a pathologic alteration of our microbiota composition, this barrier can be breached, and you can imagine that these toxins can get in the system and even some bacteria. And for that, we have the immune system, particularly the innate immune system, which will detect this danger and start a protective inflammatory response, which is not only local, it's not only in the guts. It may convey information to the brain through the blood circulation, through the chemicals, also through nervous system, the vagus nerve, for example, like we've seen, but also by directly stimulating brain cells, and specifically the resident immune cells in the brain that are called microglia. These cells, they have a particular shape. They have these ramifications because they are surveilling what is going around in the nearby cells. And these ramifications contain in the cell wall also toll-like receptor 4, which is an innate immune receptor, and in case of detection of these toxins, it will cause also neuroinflammation. Probably, for sure, all of us have experienced a group of symptoms that we can call sickness behavior when we have the flu, for example. So we feel lethargic, we lose motivation, we don't take proper care of ourselves. There's social withdrawal, probably sadness as well. There's problems in focusing and concentrating, and this pretty much resembles actually depression. And in fact, some depressed patients, it has been proven that they have increased levels of antibodies against lipopolysaccharide from gut bacteria. So the question is there a role for microbes in causing mental illness? This is in fact an old story. And I will give this example of general paralysis, which was considered in the 19th century a mental illness, and like every other mental illness, it was considered to be a consequence of depravity. It wasn't until 1857 that S. Martin Jessen found or proposed that there was an association between syphilis and general paralysis. And later on, scientists discovered that there is a direct invasion by a microbe, a direct invasion of the brain by Treponema pallidum. And so this infection was causing this disease that was not only presented with neurological symptoms, but with neuropsychiatric symptoms like mania, delusions, and dementia. So a simple treatment with penicillin rooted out this mental illness from asylums. Well, since then, such a direct connection hasn't been described, and it's not so clear for us and so easy to understand why microbes cause mental illness. But our discussion tonight will be on this current research, and we'll try to understand and propose some mechanisms. I would like to start with this study from 1929 involving 3,100 mentally ill patients, and this researcher observed that there was an excess of birth dates on the winter and spring months. And Torrey wrote in a paper in 1997 that except for infectious diseases, no other diseases had such seasonal birth excesses as clearly established as those for schizophrenia and bipolar disorder. In fact, if we think of a very seasonal infection like the flu, it is a perfect example because the flu has been associated with several psychiatric conditions. The first observation, I believe, it has been the higher incidence of schizophrenia after the pandemics, the flu pandemics in 1918, the Spanish flu. And it was Karl Menninger that said that some of these patients presented symptoms resembling mania and schizophrenia, and he was convinced that at least a subgroup of these patients had some sort of encephalitis. Encephalitis is an inflammation of the central nervous system. I would like you now to focus on this microorganism, which is object of intense research today. And this microorganism, you probably heard about it, because we have to be very careful during pregnancy because it can cause fetal malformations. Well, the life cycle of Toxoplasma gondii, this unicellular parasite, depends on a definitive host, which is the cat. And through its life cycle, the parasite will spread in the environment, and it will do so because the cat will release in the feces oocysts, which are kinds of eggs. So the environment becomes contaminated, and mice might eventually become infected. And in mice, the infection, mice are the intermediate host, is a latent infection where Toxoplasma forms cysts in muscle cells and in the brain, which were considered to be benign. Eventually, another cat will hunt these mice, and the Toxoplasma gondii infection will propagate. What was thought to be benign may not be the case, because mice infected with Toxoplasma gondii, they lose their innate aversion for cat urine and may be even attracted to it, showing that microbes may manipulate the behavior of their hosts, in this particular case, to improve their reproductive success, because these mice will get more easily hunted. In humans, this infection may also occur accidentally if we eat contaminated raw meat, if we drink contaminated water, or if we manipulate contaminated soil. And in humans, also behavioral changes have been described. This is a study that was performed in Czech Republic in the city of Prague. And latent toxoplasmosis was found to be prevalent in 10 to 20% of the healthy population, which is represented here in blue, but it was found to be 2 to 3 times higher in automobile accident victims. And this association is thought to be due to an increase in risk-taking behaviors. Infection with Toxoplasma gondii has been associated with aggression, with impulsive behaviors, with suicidality, and with several psychiatric conditions like schizophrenia, obsessive-compulsive disorder, addiction, and bipolar disorder, which was and is one of my particular interests. Bipolar disorder is a mood disorder in which patients present periods of depression and mania, but it's also a progressive disorder in which, in some patients throughout the lifespan, there's a cognitive decline and also an increased burden of other general medical conditions like cardiovascular disorders, diabetes, hypertension, obesity. So we can say it's a chronic, progressive, and multi-system disorder. It touches around 1% of the population, so I can suppose that around 5 people in this auditorium have or will be diagnosed with bipolar disorder, which is, in fact, and I don't want to be alarming, a very serious condition because it's a 4th leading cause of disability-adjusted life years in young people, and it reduces life expectancy of 9 to 20 years, and we don't know what is causing it. We know for sure that there is a genetic risk because the 1% prevalence increases to 5 to 10% in first-degree relatives and to 40 to 70% on monozygotic twins. Monozygotic twins, they share 100% similarity on their DNA sequences, but they don't share 100% risk of bipolar disorder, meaning or suggesting that there is a great role or important role for the environment. And once again, in bipolar disorder, it has been described in the northern hemisphere of the planet, an increase, an excess of winter-spring births. And influenza, in particularly gestational influenza, has been shown to be associated with an increased risk of around 4 times of having a bipolar offspring. So we explored this subject, and we performed a study in the French population. We took blood samples from 138 bipolar patients and 167 healthy controls, and we detected the presence of antibodies against Toxoplasma gondii, herpes simplex types 1 and 2, and cytomegalovirus, and we found that starting in the healthy population, 58% of the French population is in fact infected, and this is one of the highest prevalences in the world. And it's probably due to the habit of eating raw meat, of eating beef tartare, or eating non-pasteurized milk products like goat cheese in rural areas. Surprisingly, or not surprisingly, there is this really high prevalence of Toxoplasma gondii infection in bipolar disorder patients, which is 75%. So this is a correlation. It's surely not causation. So we have to try to find some pathways and what is linking these infections and bipolar disorder, with bipolar disorder. We know that the system that is responsible for protection against pathogens is the immune system, and it seems to be dysregulated in bipolar patients. Not only we see that there is an increase in the central nervous system inflammation, in the brain, in the cerebral spinal fluid, there is activation of the resident immune cells that we've seen before, the microglia, but also there's an increase of markers of inflammation in the circulation, like C-reactive protein and some cytokines. Moreover, it has been suggested, although not replicated, that inflammation precedes the onset of the disorder. So there is this complex interaction between a genetic risk, an environmental risk factor, and also a dysfunctional immune system. And I got interested by a subset of genes that encode for the immune system, and this field is called immunogenetics, because it may explain, at least partly, the inter-individual vulnerability to infection. I got particularly interested by a family of pattern recognition receptors. They recognize patterns in microbes, which are molecules that constitute them. And when they interact with them, they trigger an inflammatory response by sending this information to the DNA that will start giving instructions to the cell to produce cytokines and chemokines and other markers of inflammation, so we are protected. Two of the most studied pattern recognition receptors are toll-like receptor 2 and toll-like receptor 4. They are very similar in their structure, but they recognize different microbes. Toll-like receptor 4 recognizes gram-negative bacteria and lipopolysaccharide, through lipopolysaccharide, fungi, viruses, but also Toxoplasma gondii. And toll-like receptor 2 recognizes gram-positive bacteria by sensing peptidoglycan, other sorts of microorganisms, and it seems to collaborate with toll-like receptor 4 in the innate immune response against Toxoplasma gondii. So in another study, we sampled 572 bipolar patients and 202 controls, and we extracted DNA searching for genetic variants in these two genes. And we found that in toll-like receptor 4, there's an increased prevalence of a genetic variant that is prevalent among 52% of early onset bipolar disorder patients when compared with 37% of healthy controls. And this variant has been suggested to decrease the expression of this molecule. In what concerns toll-like receptor 2, there is another genetic variant that is being found to be more prevalent in early onset bipolar disorder patients than in late onset bipolar disorder patients. And this genetic variant has been shown to decrease cytokine production. Early onset bipolar disorder has been defined in these studies as having an onset of the disorder before the age of 22 years old. this subform of the disease is genetically more homogeneous and is clinically more homogeneous and severe. So we may propose a very simplified model in which there is a weaker anti-infectious response in bipolar disorder, and the weaker the response, the higher the susceptibility to develop bipolar disorder. So inflammation was announced as the secret killer because we don't see inflammation, but it's causing a lot of damage. Heart attacks, Alzheimer's, cancer, and also psychiatric disorders. And one of the culprits may be the pathogens, triggering this mild chronic inflammatory state. But we cannot forget that causation is not proven, and microbes are for sure not the sole cause of psychiatric illness. We have to count with genetics and psychosocial stress. Many kinds of stress, life adversities, loss of a parent, have been associated with bipolar disorder. So it's a multifactorial, and it seems to be also a multiple hit disorder in which these factors, these stressors, have a cumulative effect during their lifespan in the genetically susceptible. I would like you to focus on childhood trauma because it has been not only associated with many psychiatric disorders, including bipolar disorder, and with an earlier onset, but also with a wide range of chronic disorders in the adult, like coronary heart disease, diabetes, obesity, and so on. The link may be also the inflammation, because adults reporting childhood abuse, for example, sexual abuse, they have an increase of markers of inflammation like C-reactive protein, interleukin-6, and tumor necrosis alpha. So we try to show, because these genes, toll-like receptors, the ones I've showed you before, and childhood trauma seem to act on the same pathway on inflammation, we tested if there was some kind of interaction between these two factors, and we found a combined effect of the TLR2 risk genotype and self-reported childhood sexual abuse on starting the disease earlier. So it seems to follow a model of resilience, vulnerability, in which resilience to some stressors may be dependent on our genetics. And susceptibility may be acquired through exposure continuously to very different, diversified stressors, not only microbes, but also psychosocial stressors that will eventually crack the eggshell. In the microbes, we have to include the gut microbiota, but unfortunately, in bipolar disorder, we don't have any studies looking at the composition and differences in the microbiota in these patients and in the healthy population. I would like to finish my talk with this sentence from Leo Tolstoy, where he says that happy families are all alike. Every unhappy family is unhappy in its own way, meaning that wherever reasons contributed to increase the risk of developing bipolar disorder in a particular individual may be, in fact, particular to that individual. And it makes sense because we are entering in an era of personalized medicine and precision medicine, and eventually we'll be able to identify some kind of biological dysregulation that may be contributing and make a precise intervention in that target. I would like to thank the Champalimaud Foundation, also the Fundamental Foundation in France, the Portuguese Foundation for Science and Technology, my doctoral school, my hospital, the Psychiatric Hospital of Lisbon, and I would like to thank you for your attention. Thank you very much.
Thank you, José, for the wonderful talk. Just a small note on the prevalence of bipolar disorder here. There's enough staff at the neuropsychiatry unit to keep all of you safe, okay? Do you have any questions? There's one over there. And one here.
Thank you. I'm curious about bipolar disorder itself. How easy is it to treat? Because you give this timeline and you talk about multiple hits, and then suddenly you have the disorder, and then does it go away? So, and yeah.
So yes, bipolar disorder is a chronic condition, and it doesn't have a curative treatment nowadays. So most treatments, all treatments are palliative. This model has not been proven. It has been suggested, and research is trying to prove it. And in fact, once a diagnosis is established, there's no way to really stop the disease with the actual, with the current treatments. What the treatments do, it's slow down progression, and it may be even possible to have less and less mood dysregulation. But we are very focused on finding biomarkers that are predictive, not only of disease risk, but treatment effectiveness, for example. So you can imagine if someone has a high burden of disease on their family, if they have some characterized genetic risk, they have been exposed to some very relevant environmental risk factors. If we are able to identify some biomarkers in the microbiome, in the inflammation pathways, in other pathways, because inflammation is not the only pathway, we may have a reason to start following up these children and try to identify early signs of psychopathology, psychopathological changes. How to prevent pharmacologically is not known today.
Because it seems to be a very complex disease.
It is multifactorial.
Right? And then it seems to be a very linear approach, the one you are following, with a couple of genes, which will singly cause a change in the probability of developing it. So I wonder how much of a network approach to genes and genes interactions will you follow? Because now with bioinformatics, I would expect you to do something slightly more
compulsory, of course.
Sophisticated.
There's a lot of redundancy in these genes because many of these proteins, they have exactly the same function. We could, if we have a mutation in one of these genes, we're probably not immunodeficient because other proteins, they have similar functions. So what we have to do is study the immunogenetics in its whole and try to perform gene-gene interactions to test for important epistasis. I am currently doing this in some selected polymorphisms in the toll-like receptors pathway, but I'm not satisfied because not only it's candidate genes and candidate polymorphisms, so it seems it's not representative of the whole immune response. It's a very good question, of course. Another question? Yes.
Hello? Over here. In regards to environmental factors, how much of these are related to substances, and how much of these factors is related to like emotional or event-based influences?
You mean the weight of the
yeah.
factors?
So environmental factors, how much of it is event-based and how much of it is related to substances, like physical?
I don't know if we have an answer for that, because even if you look inside genetics and inside environmental factors, inside genetics you have genes that are highly penetrant. For example, some rare mutations, they really increase the risk of bipolar disorder really strongly. And some other variants, the ones I just shown you, they are common in the population. So they have an odds ratio of 1.5, maximum 2. And most of the genetic associations, they have the odds ratios around 2. So in environmental risk factors, the relative risk and the weight they have are not well understood. So for sure, drug abuse, if it's not a trait already, may be causative. But again, it's true that correlation is not causation. The weight, I don't know if I have an answer for it, for that. Is that your question?
Yeah, thank you.
Okay.
So it's true that there's no cure, but the lithium, for example, can help many patients, right, with bipolar disorder?
Of course, yes.
And is it known whether lithium has any influence on the immune system?
Yes. It seems to have anti-inflammatory effects, not only lithium, but valproate and some antipsychotics. Anti-inflammatory, immunosuppressive, and some antipsychotics like olanzapine has antibiotic effects. It can affect the gut microbiota composition. So there may be an effect not only in neurotransmission, but other effects may contribute to the psychiatric well-being of these patients.
So, but if it does help, if it does help with infection, still it's not a cure? Does that mean there's more than the immune? Or
yes, I understand your point, but I would like to stress that bipolar disorder or any other disorder, it's not an infectious disorder. There are infectious events that cause damage at some point. It doesn't mean necessarily that if we clear infection, we clear the damage. I don't think that is true. I don't think that will ever be true. It's just an insult in a life period.
Sorry. But has it been tested whether if you stop taking lithium, the inflammation comes back again?
I don't think so, no. Probably.
So since the event is being streamed all over the world, we have a question from one of our online viewers who asks, I wonder whether microbes can be related to different types of dementias, such as prion disease.
I don't know, that's a very difficult question. It's not my field at all. I don't know if there's any association. There's any association with between microbes and prion disease. I trust the expert of the field, and I would say not yet.
So is there one more question?
Hi, I would like to thank for the great presentation. I have two questions if you allow me. One of those is, what do you think will be the rule? You talked about inflammation. What do you think will be the rule of the anti-inflammatory treatment in the therapeutic of psychiatric illness in the future? I know that some work has been done with anti-inflammatory. What do you think? Do you think it's a plausible way of facing it?
I think it's a very
sorry. And the second question is about one thing that it's really fascinating. It's the personalized care of the, and in the clinical practice we can see that if we treat a mania and we give some drug, it doesn't work as well in some patient as it works in another, and it's kind of intriguing. And with all those variables that you put, like the trauma and genes and all that, and I know that's a difficult question also, but do you think that we can reach, by knowing these risk factors, the clinical presentation and all that, we can target more specifics for the patient treatment?
That's the point of all the research we're performing. In fact, precision medicine will evolve by better clarifying the psychiatric phenotypes. And to do that, I think we have to go back and forward on the information that comes from biological studies, in inflammation, in genetics, and in other systems, neurotransmission, the neurotrophic support of the central nervous system, in order to recreate, regroup the patients we have nowadays. And so we can refine the categories so they can be more homogeneous and we can understand what exactly we can target. These studies are very promising, the ones using anti-inflammatories and immunosuppressive drugs, but I don't think we know exactly what we are doing yet because we lower inflammation all over or we suppress inflammation all over. And one good example of that approach being not particularly good is the infliximab study in depressed patients, that in the beginning we didn't see any results, and we noticed something when we started stratifying patients according to the inflammatory profile. So those having C-reactive protein above a certain level, those were actually responding to infliximab, which is an anti-tumor necrosis factor alpha receptor antibody. Yeah.
Hi, here.
Yes.
Quick question or a comment. I heard a few years back about a patient, I think it was a 12-year-old girl who suffered from bipolar disease. And after a lot of medication, and she couldn't control her symptoms. And then the psychiatrist decided to prescribe probiotics. I don't know if this is true.
It can be true.
It did resolve the symptoms.
Some case reports.
Okay. My follow-up then would be, would you prescribe probiotics to your patients on a normal basis?
I think they cannot do much damage. And especially, I mean, there are no really side effects of the probiotics. And when you have a treatment-resistant patient, you really start to have the right to negotiate with the patient what novel therapeutics you may start testing based on scientific evidence. So yes, in that case, which is a case of treatment resistance, I would definitely try something as obvious as a probiotic. And it's a good starting point because probably it does less damage than suppressing the immune system, which has a lot of complications at the long term. Yes.
Okay, thank you very much. So thank you, Jose. So unfortunately, our evening goes now to an end. Well, at least the talks. Outside, there are still many activities. But yeah, here I want to thank definitely again to our speakers. They did really a great job. Thank you very much for coming. And please do take the chance to maybe meet them at the bar and ask them the one other burning questions you want to know about the microbiome. And also I want to thank our sponsors, Dangal, who brought us fresh pita bread we can all enjoy outside and nurture our prevotellers, I think. And Fisher Scientific also was, who was sponsoring us some petri dishes and stuff people use outside for the social activities. And then, of course, I want to thank the Champalimaud Foundation. Without their help and support, this would definitely not be possible. And I want to say also that this event was in collaboration this time with the ITQP. So we have many volunteers outside which have t-shirts with ITQP outside. And it was really a pleasure to work with you guys. So they have many different activities where we have the possibility to learn about our own microbes and also about probiotics and what microbes are used for different kind of food, what we would probably never thought they would have it. So really, please go there and talk to them. They are actually specialists in microbiology, so they will be able to tell you a lot about it. Thank you very much. And then last but not least, I of course want to thank the whole art team who is behind organizing this event and pulling this off. Especially I want to thank Marta Camacho, who was my partner in crime. It was really a pleasure to work with you. And then also the production team, Katarina Ramos, Eric David, and Gio Costa, who is the designer of this really awesome poster. Thank you very much. It was really a pleasure to work with you. And then all the other volunteers, which are outside and in between and take your tickets. And yeah, thank you very much. And I hope you have a nice entertainment now outside and learn more about the microbes over your body. Thank you.